Hi, this is Rick Powell at Nanoprobes, introducing some new products, our Click Nanogold labeling reagents. These will let you use bioorthogonal reactions to label at the electron microscopy level, bringing you closer to in vivo labeling. So you can now label dynamic vital processes at the EM level in living cells and tissues. What is click chemistry? Well, here's the original click reaction. It's an azide reacting with an alkyne through an azide-alkyne cycloaddition to give a 5-membered triazole ring, linking two components together, in this case the target and the label that you want to visualize. What makes it click? Well, it's a reaction that proceeds under physiological conditions. There's no cross-reactivity with tissues or cells. It's a fast, efficient, high-yielding reaction. The reagents and the product are non-toxic and biocompatible. And that adds up to a reagent that can be used in living cells and tissues to image life processes. Click labels are part of the larger class of bioorthogonal labels, which all use reactions that are not found naturally. They're orthogonal to the natural chemistry of cells and tissues. These include SNAP and CLIP tags from New England Biolabs, the Halo tag from Promega, or the FLAG tag, which is an amino acid sequence that's inserted into proteins and targeted selectively and exclusively by antibodies that don't recognize any naturally occurring components. Bioorthogonal labels use conjugation reactions that don't occur naturally. They use non-toxic and biocompatible reagents to give non-toxic and biocompatible products. Stay reactive during long experiments because there's no cross-reactivity and no naturally occurring reactivity. You can image a dynamic process and wait for the reaction to occur. Tag components of that dynamic process at the moment you choose to stop, fix, and examine the specimen. Here's the new products that we are introducing. The first is monoazido nanogold, which is our 1.4 nanometer Nanogold gold label functionalized with an azido group, and this reacts with an alkyne. The alkyne can be introduced into the target as a synthetic component, such as an alkyne modified nuclear base or an alkyne modified amino acid that's administered to the cells and is then incorporated by a natural process into the target nucleic acid or protein and detected by the Nanogold azido reagent. The other new product is monoalkyne nanogold, which is simply the other half of the click reaction. And that's introduced and reacts with an azido modified target, which again is made by administering an azido modified component such as an amino acid or a nuclear base, which is incorporated naturally into the protein or nucleic acid target. Features of these products are that they are bioorthogonal. There's no cross-reactivity or background binding, and monoazido nanogold will actually work in some copper-free click reactions, so you don't have to worry about cytotoxicity from the copper-1 catalyst. Stable and biocompatible. Nanogold is stable, biocompatible. It's soluble, it's hydrophilic, and it has no net charge, so there's no binding to proteins or nucleic acids that do have a net charge. It's well tolerated by living cells. Nanogold reagents and conjugates have been micro-injected into living cells without disrupting cell viability, as this paper from Belmont and co-workers has shown. Ultra-high resolution. Nanogold goes right to the target site. There's no protein or antibody or other large probe to space the label away from the target, so you can use high resolution electron microscopy to see targets at macromolecular resolution. You can optically visualize nanogold by using silver or gold enhancement, and that'll let you see it by light microscopy. On blots, this is super sensitive, and you can see as little as 1 picogram of target IgG, for example. And for specialized ultra-high resolution electron microscopic applications, we're also introducing the smaller Undecker gold reagents with the same reactivity. Applications of these products include labeling components of vital processes in living cells, allow you to ignore endogenous components like biotin and not worry about cross-reactivity. You can label subunits or molecular sites with high resolution because there's no antibody or probe to space the label away from the target. And you can multiplex your labeling by adding a click reaction to detect an additional target if there's no suitable non-cross-reactive antibody or probe that will do the job. And here are the new products with their catalog numbers and their packaging. Monoazido Nanogold has the catalog number 2026. Monoalkyne Nanogold is 2027. And these are available as single vials of 30 nanomoles, or as 5 separate vials with 6 nanomoles in each, or as one 6 nanomole vial in an introductory size. And the Monoazido Nanogold is catalog number 2046, and monoalkyne Nanogold 2047. And the Nanogold reagents are available in 50 nanomole, 5 by 10 nanomole, or single vials of 10 nanomole packaging for introductory studies. And we hope to shortly introduce Nanogold and Nanogold DBCO reagents for copper-free click labeling, so keep an eye out for those. If you have any questions or would like more information, please visit our website and follow the link from the home page. This is Rick Powell saying thanks for watching our video, and we hope these new reagents work well for you and enable you to do some new experiments. Thanks.